All right, Ice, you ready for this?
Yep,
Let's do it. There'll be a bunch of screen sharing and benchling work today, so it'll be fun.
absolutely. So if you see any struggle, that's real.
Yeah, yeah. It's a little unintuitive, I think, at first. Definitely, it took me some getting used to benchling, so it's a good time to try it out and get comfortable with it.
Yeah, I'm gonna go with the uh, general stuff first. So the cost, the logistic for today. Um,
Yeah. Okay. Mostly.
we're gonna go through some logistics. We're gonna chat about what we're gonna do tomorrow. That's gonna be very brief. We'll go through the homework question very briefly. But you can also take your time going on it by yourself. We talk about geoelectrophoresis. We talk about benchling tools, which is the DNA reading tools on the cloud. We'll get back to the gel a little bit on trying to see if we use the geoelectrophoresis as a way to do art. Then Ronan will show us how the website works for the gel art that we were working on, and we'll do some other live demos on if we want to get a new DNA, if you just have protein sequence, how do you get it, and how do you do the order on the system. So that's the overall thing we're going to talk today. It's supposed to be roughly 60 minutes. We may
Yeah.
go a little bit over, but not too much. So you can take it away for the
Sounds
logistics.
good. Yeah.
Have
So
you hit the recorded?
recording is on, so that's good. Everyone here, please do the attendance form as usual, same link. And yeah, tomorrow we'll do lab from either 10 a.m. to 1 p.m. or from 2 p.m. to 5 p.m. So whatever works better for you. You can choose whichever one is better. And there will be no lab on Friday unless you want to hang out with TAs at the lab.
Is that like what's the time again?
It'll be 10 a.m. to 1 p.m. or 2 to 5. But I will be there basically the entire day. So
One other quick update to week 4 and week 5 of the protein. We can keep them as one week too. So that way it's
great.
like everything's just like except for those.
Yeah, I know some people were clarifying in the Slack with me like what days the labs will be. So we'll also keep the protein design weeks to 1 day labs as well. So yeah, really it'll just be Gibson assembly and the CRISPR lab at the end that are 2 days. Okay, Zoom's good. Off to the races we go.
Let's go. All right. So for all of you, both those who are here and also those online, I think you've been listening to the lecture that is focusing on DNA, like how to read, how to write, how to edit. So these are some homeworks that will guide us through how do we, like learn how to work around with the DNA. And these questions are just going to be separated into three things. So I just want to clarify some of the questions briefly. So on the read part, we're asking you like what DNA would you like to sequence and why? So, it could be anything. So you can see like, some description here. Just be creative. You don't have to stick to this DNA until the end of the class or anything, just give it a try. We just want to make sure that you know how to retrieve, some of this information. And assuming that you have, these DNA you want to sequence, what kind of sequencing method would you like to use? So these are some, like multiple questions if we want to ask as the following questions. So just in case, you got stuck or you don't know what does it mean, you feel like you need to take more time to go through it. I would say, just like type it out yourself and see how far you understand. It's not like if you don't get everything right, it's going to be the end of the world. It's the first few homeworks, right? So it's, prepare just so that you can understand how these things work like properly. And then, it's a similar question like, what do you want to write? And, you may heard from George and from others as well, you can do a lot of writing of DNA these days, similar to how you write your name or whatever on the paper. It's pretty fast and much cheaper these days. So you can do these using different types of technologies. And Emily has been, showing you some of the twist capacity to do the DNA synthesis. So I think these are going to be another good homework to go through. And please, try to spend some time chatting with yourself. What would you like to answer on these following questions? And at the end, the edit is a little bit special. So on the edit, it's mostly about like something that is already functioning. and you're trying to change the function slightly, not trying to go too far so that you change the whole thing. For example, you have a car and you want to make it go faster. You still want to make it four wheel, right? Something like that. So on these, there are also like some specific and very precise DNA editing out there that you can explore and choose as a technology you want to perform. So, give it a try. So these are homework questions. If you have any question particularly about like what does it mean, why are you giving us a long text to go for the homeworks? Technically, just focus on the bolded ones, right? And like share with us what would you like to, if there's any confusion, just reach out to the TA. For those online, the note leaders might be the best person to respond to you in the proper time zone. All right. So these are some papers that might be relevant to what we are talking about, relevant to the homeworks. So if you have time, these are all good homeworks to look into. And for the DNA, there are going to be a lot of processes that we're going to try to do in silico, and you'll be doing them in the lab as well, both in the copy and join, or like doing the actual stitching of the DNA together. So these are some keywords that is going to come over and over when you're working with the DNA, which is going to be translated and transcribed into protein and DNA, vice versa. So when we work with these DNA, most of the time we have a lot of DNA, and there might be a way that we want to separate or analyze how many pieces of DNA out there are in our sample, or how do we separate them out? So you'll be doing that in the lab as well. That like for example, if you do what we call gel electrophoresis, is by putting the putting the DNA onto the gel. There are some wells up there. I'm not sure if you can see it. Maybe like Zoom was blocking it, but there's a hole, and you put your DNA in there. Then if you input the electricity, the DNA that was negatively charged is going to move from one end to the other end, and the one that was bigger and smaller would just travel at a different speed. So that's how we can compare them with the ladder that you see on the left hand side. So we go to the specific one later on. Apart from separating the gel, there are also method for making or separating the DNA pieces. Because if you have one piece of DNA that are like 10K in length, and you want to know if that's the right DNA, let's say you have two tubes, you want to know which one is the one that you think is sample A and which one is sample B. They're actually like 10K in length, both of them. You can do this kind of like restriction digest, which is the technology that came out in 1975. That's when we started the biotechnology for real. And by cutting this, the restriction means, it was restricted to cut only at this sequence. So you have these 6 nucleotide sequences going to get recognized and cut in 2 ways. There are some that will be cutting and give us the sticky end. The sticky end is the part that will be exposed and then pair with the other part, because the DNA will just try to find each other. And this blunt end is something that can bind to anything. So there are some pros and cons when you're trying to do this kind of cloning. So, most of the time we consider these endonuclease or the digestion enzymes as a scissor, where they just go find a specific thing and just chop it off. So we're going to go relatively quickly just so that we can see what is the exact sequence they're recognizing. So you can see the DNA, we're going to reiterating these kind of sequence all over the course, but these A, T, G, and C chemical structure are what the enzymes are recognizing. So if there are, a sequence of 6 of them that have these shapes, they're going to form into a particular structure, so the enzyme can specifically bind to this DNA and do the cutting. So let's say if the enzyme is supposed to be cutting to AGG, CCT, something like that, and then one of the base was changed, then it cannot bind to these anymore, because like you can see the cytosine and thymine were slightly different. And even though it looks slightly different on the paper, it's very different in the chemical chemical structure in the 3D. So this is the actual setup when you're doing the gel electrophoresis. You're gonna have a basin of the place that you can put the gel in. You're gonna put some liquid that is conductive there. Right? If you just put water there, That's one of the simple mistake. If you just put water and try to run it, keep in mind the water is not that conductive, so the flow of the electricity is going to be very slow. So we have to prepare a proper buffer so that electricity can flow pretty good. And then the DNA will just flow, in the direction. And with these bands, most of the time, the shorter bands will flow faster because they're lighter and they can like move faster through the gel. And the longer band will just stay up there. It depends on the percent gel a lot. Or like, if you have a lot of blockage in the road, you're gonna go a lot slower. So if you have a higher percent gel, most of the time the gel is gonna migrate like a lot slower.
And you'll make the gels tomorrow in lab, so you can play around with different percentage agarose
Yeah.
to see how it affects the duration of the travel.
Yeah, and just for those who are not that familiar with the DNA, again, we want to show here that the red part, which is the phosphate chemical structure, is where the negative charge came from. So even though they are HTGAA, they're all having this phosphate group, so they always have this negative charge. And it depends on the pH. So most of the time we use a buffer, and that was a word that we use in chemistry where we want to make something that has a proper pH because we buffer them. All right, these are some information. I would suggest you to take a look at the slide because if you keep like talking over and over, it's not gonna get into the real thing. But you see some suggestion here of like if you have the separation of whatever bases, these are some suggestion for the percent gel that you should be using. And again, it depends on the charge, the mass, and the length of the DNA that you should be considering when you are doing this analysis. So this is a very good video. I would suggest everyone to watch it through. It's 5 minutes video. We're not going to go through everything, but the first 30 seconds is going to be very useful information for you. So I'll do a little bit of skipping.
Is the standard laboratory procedure for separating DNA by size for visualization and purification. Electrophoresis uses an electrical field to move the negatively charged DNA through an agarose gel matrix toward a positive electrode. Shorter DNA fragments, called bands, migrate through the gel more quickly than longer ones. You can determine the approximate length of a DNA fragment by running it on an agarose gel alongside a DNA ladder, which is a collection of DNA fragments of known lengths. In this video, we'll show you how to prepare an agarose gel using powdered agarose and buffer. Then we'll show you how to load your samples into the gel and perform electrophoresis to successfully separate your DNA bands. We'll also give you some tips.
So those are almost everything that they were showing you, and they're going to go into the specific steps when you're trying to do that gel electrophoresis. So I'm going to skip to some of that.
I just want to point out, like, notice the annotations on the side here. Whenever you run a gel, it's super important that you annotate it, so that we can interpret it.
Yeah, so those are what we call the ladder, because it looks like you're climbing up the ladder. But actually, these are the DNA of the specific length and concentration, so that that like if you have the band that was close to 3K, then you should be looking for something that was around here. It may not be exact. Not all DNA are the same, but DNA that are around 3K should migrate around the 3K. So let's go to this one. So when you make the gel, after you weigh the gel, which is agarose, it's similar to like how when you're eating like the jelly, whatever snack that you had, it's actually edible. It's from algae. So you can take it and you can put it into a microwave.
Overboil the solution,
and
as some buffer will evaporate and will alter the final percentage.
Yeah, and it's gonna turn into like a clear liquid like this, and it's gonna come back into an agarose, a gel, like the candy, after they cool down. So if we skip a little bit and go to this part.
Be careful when handling the flask, as the solution will be hot. Also, be careful when swirling the flask, as eruptive boiling can occur.
So this thing is something I want to show because you have to be careful with these boiling liquid. Make sure you use the proper glove so that you don't burn your hand. If the liquid is like over boiling like that, try to put like some paper towel on top. This is like a very good video to show you how to stay safe. Again, we are trying to do a lot of learning and trying to go fast, but safety is the most important thing. Sometimes you do any mistake in the lab, you may get trauma and you may not want to go back to the lab again. And that was very bad for everyone's learning, right? So The other thing I want to show here is that when you're preparing the gel, you have to add
0.5 micrograms per milliliter.
This is the dye.
Milliliters of our lab stock to 150 milliliters of agarose gel.
So you saw that like a tiny liquid, a tiny liquid that was added in. That was the DNA dye.
The gel solution.
So
the concentration between 0.2 and 0.5 micrograms per milliliter. We're adding 5 microliters of our.
yeah, so that small amount of dye is what's gonna go into the DNA and try to bind. And once you shine the light onto it, it's gonna be fluorescent. Think about when you watch the the movies when there are crime scene, there was forensics trying to find whatever the blood stain, whatever whatsoever. If you shine the UV light or the blue light onto something that has the fluorescence capacity, it's gonna shine up, right? So this is the same thing, but the DNA by itself is not gonna shine up. So we are trying to add some molecules so that it gets into between the DNA and shine up. So what you see is not going to be visible at all if you don't add this small amount of the dyes. Very important is like don't touch it by your hands because it can bind to DNA, so you can bind to your DNA as well. So they would call them like cyber safe, whatever. It's safe, but better not touch it, right? All right. The other part that I want to show is very important is that like
so the gel doesn't pour out.
Yeah, you don't try to make sure that when you pour it down, don't let the liquid go up. Otherwise, like the gel prep that you're doing may not have the proper size of the well that you can load all the sample in. And another very important thing is that you see what we call the clump. The white thing is what we call the clump, where you just put it in so that there's some, empty space that you can add a sample in. If you forget that step, then you can't load, right? There's an easy fix by just taking the gel and just like boil it again and just like pour it back again. It's not the end of the world, but you may waste like 20 minutes when you're waiting for the gel to dry. And I think that's almost everything.
Push away any bubbles in the solution. Let the newly poured gel sit at room temperature for up to 1 hour until completely solid, and then fill the gel electrophoresis chamber with the same buffer you used to prepare the gel.
Yeah.
Pour until the gel is covered and make sure that the buffer enters the wells.
Yeah, this is very good. Like note that like you have to pour the buffer, and also when you prepare the gel also need the buffer. They have to be conductive as well. Again, if you use water to boil the agarose, it's gonna work, but it's not gonna move. And try to cover everything so that the flow was consistent. That's the gist of it. And they're gonna show you how to load the gel. And this is a delicate process.
Sample went into one well using the ladder as a reference.
You will see that
your results may be very difficult to interpret.
When you're trying to do this pipetting for the first time, it's gonna be challenging because you're doing a tiny pipetting, and if you're not used to it, it may not get into the well and it may not stay into the well. Like imagine you're doing the pending for the first time, when you were very young, the straight line as it goes straight. Similar here, if you're trying to add into a single well and just get into the well was like adjacent to it, just sacrifice that well because like you're trying to add the second sample into that well, everything would be squishy. So, they show you a very delicate pipetting approach. Is like don't expect yourself to have that steady hand for the first time when you do the pipetting. There's some tricks that TA are going to show you. So try to go to that level, but don't force yourself to fall. All right, I think like these are good example that we are looking into. This is sorry, I want to show you the picture of this power supply. That you're gonna put like
always run to red. Most gels are red.
You have to make sure that you put the red line onto the red socket and the black line onto the black socket. Otherwise, it's gonna run up, run to the top, and the length that you expect it to have for maybe like 10 centimeter, you have just like half a centimeter. Everything just fall into the water. So that's another important thing. All right, I think this is a good video, and we spent good amount of time just to show you what's gonna happen for real. So let's take another step back until like before we do this actual step, we probably want to know what is inside the DNA that we're going to load into that one. So I'm going to take a step back a little bit to introduce you two tools, which is an online tools, and you should make an account for these services. Make sure you go to Benchling and Twist to make an account. For Benchling, everyone can have an access to our shared folder so that you can get to see like what is the available DNA design that the TA have been working on. For Twist, it's going to help you prepare the synthesis of the DNA that we're going to order through Twist Bioscience. But you may not be the one doing it yourself because there's a lot of payment, blah blah blah, and the TA will take care of it. But by doing all these steps, it helps that the TA will not mess up your design. So these are pretty important steps that you should do. So these are what you need to know. We're gonna do some copy paste. We're gonna do some import from the database. We're gonna check some in silico research and ligation in the Benchling, and also like see if we can do the PCR and the cloning wizard. You see some of the cloning words coming up again and again. For the twist, we can see if we can insert and analyze the sequence, do some codon optimization, and maybe like navigate through different DNA synthesis, and also like trying to download the actual design to take it back and put on Benchling or put into some other website so that we can see it later on. And the TA can also see it, right? Because it's going to be hard if everyone has to share the password and just log into the system. So today is going to be a very brief version of the bootcamp. Of the Benchling guideline. We had a bootcamp like a few weeks ago, right? And we spent an hour walking through the Benchling design. And there are like a longer version from the previous year and also a shorter version from this year. I put the one from this year right there is around like 40 minutes, and the one from the previous year was like an hour, like a full hour. So it's like a little bit duplicated content. But if you get confused, just like go to the longer one and see the slide and other things. So we're gonna shift to the browser in a minute, but I want to show you first that once you once you have your Benchling account, if one make your Benchling account. If you're here at MIT, we want to advocate you so that you can join these How to Grow folder that we have an organization so that you can see a lot of the stuff. Others who don't have these Benchling system set up can also see it, but you may not have the ability to edit or put things in or do other things. We try to make sure that all the MIT students have the access to these so that we can share the DNA results or the DNA information easily. Take a step back a little bit. This is a slide that I always use when I talk about why do we care about DNA. And the reason is because the DNA that you are trying to work on is the core information that is going to be transcribed and translate into the protein or RNA that will function in the cells. The DNA, most of the time, is not the main thing that we do the function. So if you compare to computer system, it's like the storage system, the information that was stored in the hard disk. You have to pull the information from the hard disk to RAM and run the application. Otherwise, you won't have the actual function of whatever software that you have. So it's kind of like similar. I'm not sure what's going on with this. It's better to like when you're trying to do whatever coding. If you want to develop an app, you have to do the coding in the back end, right? So you have to do it in the DNA as well. If you want to change or make a new proteins, you should be doing it at DNA. And most of the time, you do the editing at the plasmid level because plasmid is a smaller form of DNA. It's a circular DNA, and it's very easy to work around this plasmid DNA rather than doing the genome editing and engineering. So if you go online and look at the DNA file, there was a file that has .FASTA, depends on how people call, and the GenBank file, which is where people develop this kind of system. You see this kind of format. So the format of the FASTA file is just plain DNA sequence. It would be only like HTGAA, whatever is there, and one of the more sign. I don't know what does that call properly, but that's going to be the name of the sequence. GenBank, we have a little bit more information where we have what is the size of this DNA, what is the annotation of each of these DNA, and you can see that which file is going to be bigger, and also which file is going to be more important when you're working with it, because if it asks you, hey, can you go and look for the gene first and protein GFP from this file? It's probably pretty hard for you to figure out where is the HTG, and which of the codon will translate into whatever amino acid. And at the end of the day, there's like 1,000 amino acids. It's going to be very hard for you to figure out where your protein is. So that's why these program are very important. These are some examples that I think our TA has been exploring into how to get this information. So if you go into the NCBI website and look into this kind of like chromosome information, enzyme information, blah blah blah, you see this kind of information out there. You can also go to UniProt, which is prot stands for protein. Where can you get a protein information? In this protein information, there will be some sequences option, and you can look through what is the source of the DNA out there as well. So these are some kind of like database that you can look into. Again, back to this, we want to make sure that we emphasize the direction of the DNA. Again, you see that in the FASTA file, they have only one line of like DNA, right? But DNA is usually in a double-stranded form. The reason that the FASTA file can show you only just one of the strand is because the DNA has the directionality. Like if you have the 5' like from left, let's say, to the right, you can actually make a copy of that one in the program very easily, because all the A, which we have T on the bottom, all the C, we have G on the bottom, and vice versa. So that's why the convention of the 5' to 3' end is very important. Again, 5' go to 3' is usually on the top strand. If you look at the bottom strand, it's like rotating. So the 5' is on the right-hand side on the bottom instead, and going to 3' on the other end. So this is like how it looks when you go to Benchling. You see some of these screenshot. We'll go to the actual program as well, but you can actually do the import of these DNA. You can also make a dual DNA or an RNA sequence. You can do the restriction digest, like which we share about the digestion a little bit, right? That you can find where you can do the digestion of these things. And if you show like equal R1, for example, they will show you how many sites that would be cut in this actual DNA, something like that. And this is the Voyager gel that Benchling actually show you. They will show you what is the size of the the pattern of these DNA digestion. They will show you after you do the actual digestion and run to the gel. You can also choose the different ladder. Right there, you can make different pattern if you use different restriction digest enzymes. And let's do a quick kind of walkthrough right here. So I think this slide is shareable, so I'm going to go into this sequence right here. I'm not sure if my Zoom is blocking things, but yeah. Do you still see my screen up there? Yes.
Yeah, I think we got it.
Sure.
We're
Cool.
good.
So the link that I put up there is to go into the NCBI library to look for the phage lambda chromosome.
See if y'all can follow along with this, because it's good to know.
So again, if I don't have that link, how do? I go to NCBI phage lambda genome, and there'll be some information show up. The first one on the top, it might be a little bit biased because I search DNA on Google every day. Like if you don't see it on the top one, it's not your fault. But yeah, if I click into this one, it's showing me actually the same thing as I put up there. You see this number, this GenBank extension code, which is J02459.1. Dot is like a version of it. Sometimes these DNA sequences were sequenced in the 80s and 90s. Maybe we need to update them because the quality of the information was not the base, something like that. So you see .1 or dot something all the time. If you take this information and put onto Benchling, you can actually download all of these, information, whatever. There's a lot of them. And these coding sequence that has like the protein, we will translate into the actual DNA. This is the actual DNA. Again, I don't think any human can, if they don't have the spatial power, can extract the information from these kind of like long strings of DNA sequence without the computer. So let's say going to Benchling. And you see this one, the one that has like this logo. Just get into it.
They ask you for like affiliated with the lab. Is there like something for the program? And then
HTGAA.
you found it?
Yeah.
Okay.
There should be one.
Yeah, there should be.
Yeah, so there should be an organization if you can find it. If not, you can also join the organization later.
Yeah.
So if you don't have an account, you have to, make your account. If you have one already, just sign in. I'm gonna use my not frequently used account just so that I don't have a lot of things showed up when I try to search for things. But for this one, if I go into the folder that I have access to, I'll go to maybe HTGAA _ICE, which is my folder when I was taking this class 2 years ago. I can go to this plus button here can just give you, a new folder, a new entry. An entry is something that you can take notes as well, because Benchling is designed so that you can do, and you can use it as a lab notebook as well. We're not going to go too far in the lab notebook, but I want to go into the DNA part. So you can either do new DNA or import, or do it from spreadsheet or something like that. Most of the time, you'll be using these first two buttons where you just do a new DNA and import DNA sequence. So I'm just gonna try the DNA sequence first. And just say like gibberish, whatever. And they are gonna ask you if this is a circular DNA or linear DNA. I'm gonna tell them it's a linear DNA. You can tell them what is the basis content. If you don't know, it's okay to just like create this gibberish file. It says like there is 0 base pair. I would try to type something down here. So, it's like imagine the cat was walking around. No, it's probably put some tap. It was walking around, and you see some of the red thing pop up there. It means it's not compatible with this software. So it's only ATGC and other characters that represent DNA can be put up there. Again, it may look strange to you, but some of these refer to something like it's either AOC, it's either AOT, something that's not A, something like that. So some of the characters apart from the ATGC can be put in here as well. You can also do a copy paste. So if we go and copy this sequence, you can do Control-Z, Control-V, and just paste. Then it's going to have an option right here. If I enter, these are the information of the DNA at the top here. You can also see that there are some restriction digests, like recognition site, that pop up here. So those are, something that will be recognized. And it depends on whether you have the right, restrict digest system in there or not. So on the right-hand side, there'll be several buttons that show you whether you're going to do some digest, you're going to do some, put some primers. Primers is a short DNA sequence. We're going to talk about that later on. Whether you have history, you have like coding background is very important. You need some kind of like timestamp. You can go back to the previous version in the history so that if you mess things up, you can go back. There's alignment to compare whether this sequence is the right thing or not, because once you make a DNA, you probably want to check if it's correct or not correct. You can do by multiple approaches, but doing sequencing is one of the process. You can also like do this kind of like change from linear to something else, change the name, change the folder, whatever, similar to how you're navigating into, your regular folder. So let's change from, this kind of like copy paste approach to another approach.
So what are we seeing over here? I mean, I pasted this, and what are those hierarchical?
Like on the left?
The tau, SSI, all of those stuff.
You mean?
Oh, the different restriction cut sites.
Oh, is actually different restriction enzyme cut sites.
Okay.
That is available in the commercial or available in the universe or whatsoever that they collected. You can change that to another set. So this is like showing all the enzyme lists of the cut sites. I can change it to, let's see, everything from NEB. You can have a smaller set, or not sure if we have like how to grow. This is like, one RCD that's Ronan. And
That actually might be our how to grow one, yeah.
yeah, and there's nothing because, we have a smaller set.
We have 10 restriction enzymes that you'll use tomorrow in the lab.
Yeah. So let's do import instead. So here, you're trying to import, you can actually download the file from this website. You can go to send to and go file and download as GenBank or FastA. It's FastA whatsoever. I don't really like this process because if I use Benchling, I can copy this GenBank code and put it import from database and just put it down there, search, and get a pop-up as this entry LAMCG lambda phase genome, complete genome is like 4,800, I'm sorry, 48,000 in length. Hit import, and then you're gonna have this DNA file. It's kind of long. You can also try the digestion that was suggested in the slide. Let's say we do equal R1 and do cut. You see that there's the cut size that was available. If you go to the button virtual digest, the tab virtual digest, this is the actual comparison. You can also change it, the ladder from whatever ladder we have. For example, maybe we have the 1 KB plus
Yes, 1 KB plus thermo.
So you see a little bit different pattern of the ladder. So this is kind of important because like if you're exporting this picture that has a different ladder and you go into a lab and trying to compare them and you just like get confused. Why is there only 10 bands on my design and the actual gel has like 15 and you don't know which one is in the order. So make sure you compare the right one. You can do it again.
How does this virtual digest work? It's like a model predicting what the experimental output would be?
Yeah, so this is just like, a Control F like search for the actual cut sites. So let's do it again. I'm going to show you to look for equal R5, let's say. So this equal R5, I'm sorry for, like you may call it equal RB, it's not wrong, but like, I call it conventionally because I'm old. So these are Roman number. So the last number is actually like, Roman number. So I call it like equal R1, equal R5. So for this one, you see the explanation right here, which is this is going to cut anything that has GATATC. So what it's going to do is it's going to search into this whole DNA sequence and it tell you that there are like these many cut sites. If you click cut and run digest, what's going to show you in the virtual digest is that once you cut it, it's going to be segmented into a different link, right? And, if you're, from, engineering background, doing sorting before, they just take this sequence and do the sort and you just show you how many bands is there. Sometimes if there are 4 bands, you may see only 3 in the gel if 2 of them were on the same size. So, in the physical world, it's not always, obvious compared to our in silico design. So, all right, I think that's, we spent a decent amount of time showing you that there's some button around here. So I'm going to go back to the actual slide just so that we continue with other information. I'm not sure if I can go back full screen here. Okay, I could. So maybe I'll give like Ronan and Alex, some time to share about this gel art.
Yeah, so you can see pretty quickly how what Ice was showing, you can actually make artwork with it. So we're going to take some inspiration from a project that was already done by Paul Vanus. And so he created these really neat designs using gel electrophoresis with DNA. And so I don't know if you see what the control is, but you can use this. Yeah, I might let you do it.
So this is the
So first and foremost, you need to learn how to use Benchling. I'll just preface this discussion with that. So ultimately, when you do your write-ups for your homework and for the lab, please have everything in Benchling. However, you'll also quickly notice Benchling is not set up for iterating quickly on artwork ideas, right? It's more for like actual genetic editing. So as part of this class, I made this really simple interface to help you just scroll through all the possible cut sites that you can have on the lambda phage genome. So if you want to pop that tab open.
this one.
Yeah.
Can I ask, when you say in the homework we need to have it in Benchling, what do you mean? Like, do we post pictures from Benchling or?
Yes.
Yeah,
Yeah.
okay.
So you'll make a design in lab this week and you'll run the gel. And then it's what is most helpful is if you have a photograph, like screenshot of your Benchling digest, virtual digest.
Okay.
Yeah.
You can also put the link to, like, you just embed the link to your website, like. Yeah.
Yeah, so at the top here, you can see like a simulated, same concept as benchling, right? Just kind of a simulated gel. And so you can see on the left, you can put a ladder, you can randomize through all these cut sites. Depending on the gel you're using, you might have different numbers of lanes. So I think I default to like an 11 lane gel or something. But you can add to that or subtract. Yeah, so basically just play around. You can use this to play around or benchling. Try to come up with some fun design that you want to run, because then we'll actually do the restriction digests, and then you'll run your samples in a gel and try to make some artwork. That's the big picture
Do
here.
we need to do this before tomorrow's film?
If you want, yeah, if you want to have more time to create a fun design, sure, but also you can do it during the lab. It's not, yeah.
Yeah, so what you can see here, one thing I want to emphasize here is that, like, let's take a look at number one. Lane number one, if you go to this nice table that Ronan organized, it says it's gonna use 1 regular of SAC1 and 1 regular of SAL1, SAL1. So if you go back to Benchling and try to do that, SAC.
What DNA do we use in the lab tomorrow? Like, what do we cut?
It's going to be the same as
Yeah,
we're
it's gonna be this lambda
lambda page. So it's 48,000 base pairs.
The worksheet digest will look like this on the second one. So this is like, like what Ronan put into this automation website. It's the same thing as Benchling was showing you.
So what I'm showing here is like in this table is the quantity or volume that you'll put into a PCR tube, and then you'll incubate that. And then you'll take some of that out, probably dilute it, mix it with some loading dye, and then run that in your gel.
Yeah. So if you have no clue what is the enzyme, which enzyme do we have? Just do this randomize and see like which pattern you may like, and just go down and see like, oh, it's lane number 5. Lane number 5 use equal R1 and banish 1 to do this. Another trick, because like if you start doing this for the first day, you may not realize or recognize a lot of these enzyme names, which is fair.
And then for any nodes or anyone else using this website, you can turn on and off the restriction enzymes if you don't have all of these in your lab.
Yeah. So let's get rid of equal R1 and let's show only 4 enzymes. Then do the randomized. Then you get, not as fun.
Hey, you can still make something with that though.
Yeah.
Yeah. Anyway, just something to play around with. Just want to show that. So you have that as a toolkit. Yeah, for tomorrow.
Right. And Alex will be, one of the lead TA to walk you through the artwork tomorrow. Right. Is there another thing that we want to show? Yeah.
Yeah. So quick overview of the lab on Thursday is you guys are gonna have your designs, you're gonna come in, you're gonna make the TAE, so the buffer, you're gonna make your gels, and then you're gonna digest your plasmid into the designs you want, and you're just gonna run it. And that'll be pretty much the entire lab. And so previous examples that we've done include like the MIT, spin your eyes a little bit, but yeah. And then that was normally done by just digesting and running it. I recently ran the other one, kind of like the triangle, and that was done by just running the same size in each lane, but I just paused it after 5 minutes and put in the DNA. So I didn't put everything in at the same time. And so that's like also another option you could explore.
Yeah, so you put that
shadow here.
That's,
So you put these outer bands first, right?
Yeah,
And then
and
you
then-
ran it for 5 minutes. And then
and
you
I
put the
stopped
next
it.
two.
Yeah.
You get the best
Yeah.
design.
So there is a lot more granularity to do it that way, and you can get like much more clean and cool designs. So it's something to explore, but it's not set up in the Benchling and Roland's thing how to do that.
Yeah, I don't have a timing feature.
Yeah.
So one thing I want to add in, you see that, that shadow things that show up here, that's not DNA, but it show up in the gel when you take a picture, because that's the physical dye that you dye. And the reason that we have that is because we want to make sure that these are usually the dye that was like will migrate faster than your smallest DNA. So that if you know that these dye just go off the rail, you just stop running the gel. Otherwise, some of your DNA may migrate too far.
Yeah.
And you'll see most of the gel running will have some color in it. That's the reason. And another reason is that the color, the water, if you put water into water, it just like diffuse, right? So these will have some glycerol to sort of like cover and coated them. So that when they fall onto, the sink, if you put something that's way more heavy onto the aqueous solution, they're gonna fall first and they will diffuse later. So that's why we need to put some of the glycerol content into these liquid when you load the gel. Just follow the instruction of the dye that was coming with the kit.
All right, and then once you have your images, whenever you post it on your website or either this lab or future labs, this is examples of how it should be labeled. So you don't need to do everything in
This
the
may be a
red,
little overboard, but
but
just to give you an idea.
as long as you have the ladder labeled and the sizes on the ladder and what is in each plane, that should just be enough. And yeah, and now I'll pass it back to Ice for the live demo.
You see a worse version when I share mine.
I will say though, like just like for the first few labs, it might be good to like
Yeah,
what's called overdraw, like over label. Like, can you go back one slide again, Scott? Like for example, even here, like you can see there are little dotted lines like in front of the ladders, like where the hands are, the regions are. It makes it a lot easier to quickly see and understand, especially again, since some of you might be doing it for the first time. So definitely go on the side of like over like drawing first and then later on the tug.
Yeah, too much information is always easier to resolve than no information. Right?
this is actually a gel that I ran for your Gibson assembly lab. Me trying to figure out how to design the primers. So you can actually see like MUAV is the plasmid that we're using, and then we're PCRing out the whole backbone, but not a certain chunk.
It's not as clean. You see, like some pieces are giving like other bands that you may not want.
So what I was trying to aim for is like to know that this is a smaller band than this, so it is shorter, right?
Yeah.
Anyway, we'll talk more about that later, but.
All right, let's go for another demo. So it is like another real demo when you're trying to, let's say, convert your plasmid that is expressing one protein to another protein. So I'm using the fluorescent protein as an example here. So what we're gonna do, like in silico and also maybe in the lab if you have time, is that we're gonna take these DNA, a plasmid that has GFP coding sequence into it. So when you put this plasmid into the bacteria, it will be green. I want to cut this GFP out and then go and grab, a red fluorescent protein sequence from some other places and just like put it into this site. And once we assemble them, it's going to turn the bacteria from being green to being red, right? So the actual, I'm going to show the actual physical picture later, but let's go to the Benchling project first. So most of the time when you do this process, you need at least 1 or 2 template DNA. That's what we call. You can actually PCR the whole thing. PCR is a process where you have, DNA primer that was bound to the left-hand side or the right-hand side. So like identifying where you should start taking the piece of DNA out, or you can just do the restriction ligation that we just showed you. So let's take a look at the ligation folder in here. You're gonna get that out. So I'm gonna start with this, DNA number 1, where I'm gonna take this, this piece out. I know that this SFGP, have some restriction digest cut site around on the left and on the right of it. So we go back. It says that I should cut at maybe PAC1 here and MSS1 right there, or around that side. So we go and cut around PC1. How to grow doesn't have it. Let's assume that we have it. There are other options. So you're not always like restricted to use this thing. Let's use PAC1 and MSS1. And it just do the digest. When it do the digest, this is the Warsaw digest that you see, right? There's a big band and there's a smaller band. The bigger one is around 2 kb each. The smaller one is 1 kb each. So you probably want a bigger one because, according to the picture that I show you here, the backbone is bigger than the coding sequence, right? And if you click into the backbone, it's going to be highlighting the part of your plasmid here. And you can see that it was highlighting everything but the SFGP, the green part right here. So if I do copy, like right-click copy, you can also do Control-C and go to new DNA sequence. just like digest. Make sure to call it linear, because when you cut them, it's changed to linear DNA already. And then do the paste. And you have this linear DNA that have this MR and CoE1. These are the selection marker and replication part for their plasmid. So those are important things. So if you want to put the, the MRP part into it, I think I have it on the other one. So let's say I have this. It's fine.
You're new here.
It's okay. It's okay.
It's a tradition.
It's my hobby. So another thing that you can do is to do PCR. So I go to this, DNA template. One thing I'm going to show you, which is like, interesting trick, is that I want to, copy this part, maybe this part to the end, and I'm going to cover some of the other part as well. It's kind of hard for me to do it when the linearized sequence here is not, easy to navigate. So I'm just going to do right click, reindex, and tell them like, hey, can you make this number 1 on the sequence instead? So it's going to rotate a bit and move number 1 to whatever I just told them. So what I'm doing here is I'm going to do a PCR by using this primer. This primer is a single strand DNA that's bind to this side of DNA.
How did you add the primer?
When I add the primers, you can just right click and create primer. You have to tell them if it's forward or reverse though, because if it's forward, it's going to bind to the top one. If it's reverse, it's going to bind to the bottom one. Very important. A lot of scientists design it and click on the wrong strand, and it's not going to work because it's a single stranded DNA, right? It's not the same. It's complementary, so it's very different. So I actually put this sequence in already, and I know that I want to cover this whole MRFP sequence, and I actually design another primer right here. What I can do is that on this, primer button here, I can pair them. And by pairing them, it tells you what is the size of it. There's also an option to create PCR. If you create PCR, there's some options to keep use the primers, keep something. And I just tell them like, hey, just make it. Then I have this new sequence, smaller one, 1.2 kbh. That is a new DNA piece. So if you want to do ligation, I want to show you another tools, which is an assembly tool right here. Sorry, it's not showing everything. There's some cloning available out there. The most classical one is the digest and ligate. If you start, it's going to ask you, sorry, I'm going to put this up. It's going to ask you, hey, do you have a backbone part? Do you have an insert part? Similar to how we are doing here. Do you have the backbone that doesn't have, a lot of things that was just like cutting out the coding sequence that you used to have. And the insert, that's usually the term that's something that we're putting into the backbone that we used to have. Most of the time, the backbone refers to something that can replicate and still function. And by having this replication origin and the selection marker, they can still replicate, but they may not have the thing that we like, right? So here, the backbone is the first part and the insert is the second part. So from this backbone, you can either go to the digested one or go to the first sequence. And I told them, I sort of like annotate that I'm going to use MSS1 and PAC1, right? So we go to digest and just select this part. You can actually hit right here, set from selection. So you're going to have this preview showing that, hey, you have this digestion, and one of the end is having this blunt end, having just AAATTT, 2 different strands. And you have this PAC1 cut site where you have this sticky end, having this AT hanging. So if you go and choose the insert that we just make here, so this insert, push that further. If it just like click at PAC1 here, and scroll down and click at MSS1, set from selection. Seems like the software sort of tells us that it seems like it's gonna match perfectly because it has the sticky end of the PAC1 that match each other, and you have the blunt end from MS1, it's gonna match each other. Blunt end and sticky end can sort of bind to each other, but there are some extra process. So most of the time you have one sticky and one blunt, it could work. So this one says, looks like everything checks out. You can click done and give it a name. Assemble. And you have this thing, you're gonna save it into a new folder. Then you have a new plasmid right here. It also gives you the assembly history, where it shows you that you have these two pieces, the orange one and the green one. The orange one is the backbone, the green one is inside. Going back to, our slide here, when you're done, and again, like there are some sequence link up here if you're not following. You can always take a look at these sequence that I attached here. So when you're done, you're going to be running this gel. So this is the gel that I cropped from when I was doing a similar cloning. So I do the digest of this plasmid. I have these 2-ish KB, and this is like how I usually annotate. I don't care. I always use I always use one ladder, and it's like NAB 1 KB plus, and I know that this bright band is 3 KB, this another bright band is 1 KB, this another bright band is 500 base pair. I just don't label anything because I do it 3 times a day. So this is a bad example, but you do it the first time, please label them. So I know right away that this is the sequence between 2 and 3 KB. I'm going to take this one because it's a bigger piece of the backbone I want. This is the other piece that when you do the digest, is going to have another one. And you can see that the bigger band has a brighter, signal picture because it has more DNA, right? The bigger it is, the more space for the dye can insert into. So in terms of number of molecules, they're actually equal, but the bigger piece has more DNA in it in terms of content. And that PCR is usually a lot brighter. So the insert, I make it by PCR. So you get a fat PCR band. Seems like it's correct. I have only one band. So I just do physical cutting. You take the scalpel, just cut through the gel. Put into the tube. Add some buffer that helps with the dissolution of the gel. And do something similar to when we were preparing for the gel. We just heat it up. And once we heat it up, dissolve. It's going to come back, and the solution was prepared in a way that once it dissolves the gel, it's not going to solidify that easily. So once we have that, you're going to put into the column, do some purification of the DNA, and takes DNA fragment 1 and fragment 2, put them together, and add the ligation enzyme. And then they're going to connect. And when you put into E. coli by heat chop, you can do these either, heat chop process by putting into the cold temperature. The plasmid is going to stay around the bacteria, do the heat chop. There'll be some fracture of the cell membrane. Some of the plasmid will get in, do a cold chop again. It's going to come back, and the membrane is going to stick to each other, and just you plate these things onto the plate. You're going to see different color. And if you start from green, you can turn to red, and you can know if it's correct or not by the color changing. So, that's all of what we want to share about Benchling. It seems like I spent too much time on Benchling, but hopefully that's the important thing that we want to walk you through. The only thing I want to share today is about the twist system. So these are good slides. Please try to take a look at this information again. It's much more simple than what we just showed. And it's going to be about the bacteria, what is a plasmid, something like that. We're going to go very quick, skip everything so that we can go, have dinner faster. One important thing is that there'll be codon system where each of the DNA sequence will be organized in a way that they can be coded into different protein, right? So these are very important things that we have to be aware of, that there will be a 3 nucleotide codon, like AUG or ATG. It was AUG because it was transcribed to RNA already, and if it has UAA or TAA in DNA, it's going to be the stop codon. There was TAA, TGA, TAG. Those are something that will be the stop codon. If you see something that was AUC or ATG, it's going to be isoleucine instead of the methionine, blah blah blah. There's 20 of them. I can remember only a few, but if you keep working with these, you remember the start codon, because you have to look for that start codon no matter what. So these are the table that helps you encode what is a set of 3. There will be 64 possibilities, right? Because there are 4 at each of the position 4x4x4. The 64 possibilities. Some of them will refer to the same thing. For example, if you have like UC or TC followed by whatever amino acid, whatever nucleotide, it's going to be serine no matter what, something like that. So these are how the translation occur. And we'll be using some kind of a codon optimization because some of the tRNA that was carrying the actual amino acid was in different abundance. So if we kept using, the proper ratio, that we use the proper amount of tRNA, then the expression of protein is probably better. So we take that from, the frequency of these codon that appear in the E. coli genome. There are these kind of like similar thing, you transform your peak colonies, you inoculate, and if you don't want to do that by yourself, we have an option where we were supported by Twist Bioscience, where they will do the synthesis, so do the cloning by putting onto the backbone, something similar to our ligation or Gibson assembly. We will talk about the Gibson assembly later. But they can also put that into the plasmid and ship it to us as a plasmid or give us a fragment so that we can do the cloning with similar to the restriction ligation that we do. So, there are some options out here. There's a way that you can order as a fragment. There's a method that you can order as clonal genes. We support the clonal genes because when you're trying to do the cloning for the first time, the troubleshooting usually takes, multiple days because you have to wait for the bacteria to grow. So if you can skip cloning, that's actually better for your project because you don't have to spend time on something that you don't know how long you're going to take. But, we encourage you to do some cloning if you want to try. But if your question is just like, hey, I want to see if this protein function or not, just order it. And there's some adapter option. It was like helpful to, amplify things from, the system that have adapters because the synthesis system sort of work that way. So, you can get into, the homework too and see what you want to synthesize, blah, blah, blah. If you go through this slide deck, it will show you how to synthesize the L protein. I'm going to show you some kind of like demo of taking some fluorescent protein. So, I'll go through the system first, just for the slide first, and you go into this slide, you just create a new synthesis of the gene. You can choose whether you want to go for the clonal, the gene fragments, or the library. The library is not quite our option because we want to have you go for something that you design exactly and not trying to try too many things because you don't have a lot of time in the lab. So it's either the left or the middle one. Go for the left one. So that they spend 3 days working for you rather than you spend 2 weeks trying to troubleshoot. So you can either give them the actual nucleotide sequence or give them the amino acid sequence that we have to reverse translate into the DNA sequence. You can just upload the file, do the manual import or whatever. And there are some codon optimization tools out there as well. If you can't find these in Google, just log in once you have the account and just go to resource and scroll down, you see codon optimization tool right here, like on the bottom here. And just put it in. What is the name? What is the ORF? The most important thing is that you have to put this information in, then they will calculate if this DNA can be synthesized or not. What does that mean? It means that some DNA are actually very hard to synthesize. Let's say that you want to make like all A. It's very hard because you don't really know, how long the A is going to be. There's some like reading or some processes. It was very hard for what we call homopolymers, where these, repeating units is kind of hard to count, whether we have 8, 9, something like that.
Does anyone remember what Emily said yesterday? Out of curiosity, because I know she mentioned very briefly in the lecture, like which ones they have difficulty actually synthesizing, especially.
Yeah.
I was going to pay attention much.
Yeah.
They have a hard time synthesizing G and C. So if you remember, they were talking a lot about how they try to extend their range and how many G's and C's in the lower end and the upper end. So this is something else to keep in mind too, but not to do that.
Yeah. So it's all GC. It's kind of hard. It's the bond that was binding was so strong because they have 3 hydrogen bonding, right? Is the GC content is not too high, around E. coli, 50%. It's going to be easier. So, whenever you have extremely high GC content, it's crazy. Or when you have a lot of repetition, it's hard to make sure that it's correct because trying to see if it's like 18As or 19As. We're fairly difficult when you're looking at the curve that they're all like the same signal. All right, so these are what you can do for the codon usage or whatsoever. I'm gonna go to the actual demo after we finish the whole slide. I think we did. All right, so let's go to Twist. Make sure you go to Twist Bioscience. There might be some other company out there that has similar names. Make an account if you don't have one. And technically, what I want to synthesize, let's go to FPBase, which is a fluorescent protein database, where you can look for something out there. I'll just search for a random thing. GFP, there's a super folder right there. There's a triplase GFP right there. I don't know what that is. Let's give it a try. If you click onto this triplase GFP and scroll down, there'll be a protein sequence right here. From that protein sequence, you can actually try to convert this protein sequence into the DNA sequence from the codon optimization or whatever tools. So let's see, I go to resource, codon optimization. Oops, page not found. Sorry, Twist. I'll go through another route. I think that link was broken. So I'll try to order it state. I know this because there was another system that I have been using before. So I'll go through replace. And you see this kind of struggle all the time. I didn't do this beforehand, so I didn't know that the link was kind of broken. So I take this DNA sequence, this amino acid sequence, and it's gonna load. The app is a web app. I'm gonna tell them it's a clonal gene. I'm gonna give them amino acid sequence. I'm gonna do manual import because I have only one sequence out there. Tell me that if you're trying to do this amino acid sequence, you have to make sure that it's one of the 20 standard amino acids. So if let's say X is not part of that 20, it's gonna spin out some error. And this star marks the stop codon, and then don't add stop codon manually. So this is something you have to make sure that you add it in. And this is an instruction that based on the codon usage table you choose, the most frequently used codon will be selected. So I type it here. I call it triplase GFP. We give the name. One thing that I want to add in, very important, the space is fine, just ignore it. I want to add a stop codon as well with the star there. I'm going to use E. coli, Escherichia coli. You can also choose another one, Homo sapien. Yeah, so choose human as well. So
Yeah.
Does your codon optimization differ between organisms?
Yes, yes.
Okay.
So
how do you select that
if you want to do it in the How to Grow lab, we suggest that we do codon optimize for E. coli because E. coli is the easiest organism to work on. But if you want to work into the mammalian cells, where it's a lot more complicated, you have to clone in E. coli first and then try to extract the DNA and throw it into mammalian cells. Injecting it into your cell is not going to work. So you have to select the vector. If you have, some option right here is add a cloning expression vector or just regular vector, you can just choose from here. So let's say I was choosing a Homo sapien codon, right? So I'm gonna say let's use the P-twist CMV because CMV is a promoter that works for mammalian, something like that. It seems like that the web app doesn't show me the further one. That's fine. I'll just use the cloning one. Let's go for P-twist app. So go, and then it's gonna start analyzing the complexity. You see this complexity. My screen is not that big, but it's gonna go and analyze the complexity and see if it's standard or not standard. So
The complexity of what?
the complexity of DNA they're trying to synthesize. Yeah, if it's complicated,
Of the
it
optimization.
may fail. Yeah.
So it's not related to your sequence, so just how big you make it.
Yeah.
So.
So I can download this as FastA, CSV, GenBank, even download it as GenBank, which is already downloaded.
Just because it's difficult to synthesize doesn't mean they won't try.
Yeah.
So, yeah.
It's just that, like, you know, if they say like you're gonna get it in 5 days, then we'll be like, actually it's kind of hard. We're trying to troubleshoot this, something like that. So I have that, and I'm gonna do the import, and I'm gonna choose the file, and it just download from Twist. The replace. And what you have right here when you just upload it is going to be this thing, where this is the vector backbone, right? And this is the insert. So again, we can try another thing. This is the amino acid sequence that we just get, right? We take this and go here and try to search for it. You don't really see it because these are DNA information, right? You're just dropping the amino acid sequence. If you click here, amino acid, it's popping up here. So that means that this is the real sequence that was converted into the DNA sequence that is going to get translated already. You can do the right click here and create translation, and it's going to give you replace. It's going to give you this KSKGEELFA. So it seems like it's the correct one. Very important, you know, check if the protein is going to express properly. Anyone spot one flaw of the design I just did? Very important mistake.
You didn't make sure it would be inserted in the right orientation, maybe?
That could be an issue if there's a promoter there, right? So I just like want to synthesize this DNA. So there was one thing that was adding extra when I copy paste this, you know, amino acid sequence. I add this stop codon, which is TGA, right? So if you do the translation, it's gonna give you this like star, whatever, right there. Oh, someone was drawing on my screen. This is not a mistake. I did that because I make sure I need the stop codon. What is the mistake?
Stop codon?
Yes,
Mhm.
I forgot the stop codon, the start codon. So if you have this, it's not gonna express. It didn't have the start codon. So you're not gonna have a protein. You want to fix it, you can do, you know, add it by adding to the primary that you're going to copy into the other spot, because the primary you can extend it a little bit further so that some of the DNA sequence was included into the primer or the new DNA that you're going to get as well. All right, seems like that's a, you know, good showcase. Maybe one thing I want to show you is that
How expensive would it have been if it was like under the limit that they have, like under 500 BP?
oh
Does it like dramatically go down in price if it's under 500?
yeah, so there's some difference price bracket. If you go below 500, I think it's gonna be assumed to be 500 base price all the time, something like that. And it just depends whether you order as a clonal gene or a fragment, because for the fragment, just like make a fragment and give it to you. If it's a cloning, they have to put into the backbone and give you the assembled backbone. That means they have to do the sequence verification for your stuff as well, also taking longer.
You can expect it to be roughly like $50 to $150, so it depends really well.
Yeah, there's some price bracket, and that's why, like, when you're going to make a library where you have a lot of the library members, it's going to be a much cheaper price per like identity, right? So let's do one quick thing. If we do copy paste again, but I want to copy paste twice. To click this. So if I apply here, I'm assuming that it's going to be a little bit more difficult because I give them a repeating sequence. Error. Okay, let's see what's the error. Invalid characters. Oh, oops. What is that? Oh,
So that's not the error we were hoping. to
That's
find,
not the error we
okay,
are
but,
showing, yeah. So this is like I was in the genes and I was adding as DNA. They assume that everything here is going to be DNA ready. Seems like they skip autocodon optimization. So if we go back and do the order again. Sorry. You have an active draft, you haven't ordered yet. Monogenes, amino acid. This is the reason why that one was failing, because they don't like that I was putting amino acid sequence in the DNA order. Continue. Oh, I have to take the couldn't use this. Let's do E. coli.
And you guys will have plenty of time to do this in the homework too, because it's designed just so that you walk through this, so.
We don't add subcodon automatically. If you need it, add it. Yeah. Also, they didn't say that they're gonna add start codon, so make sure you add start codon. All right. This is a sequence. They said complex. Let's take a look. High repeat density detected, repeat region detected. So if you get into this piece, you say like, this is a repeat region, repeat region, repeat region. So even though we tell them, hey, do some kind of optimization so that we can synthesize this DNA as good as possible, they just like return something that has like duplicated DNA sequence. And by having the same DNA being tandem, make the repetition of the DNA, the codon, more frequent than not the same amino acid, right? So, if you're trying to make a concatemer, like a repeating unit of whatever protein, it's usually really hard. And that's why the spider silk is very hard to make, because it's always a repeating unit of protein. All right. I think I'm going to stop right here. Any question? Sorry for taking a little bit longer, but I hope, the walkthrough is useful for all of you.
Please try to blow our test budget. I think it's probably one of the best opportunities we have with the course, especially because it's so difficult to get these in general. So the fact that you can just spend it on a fully assembled DNA to express something is quite frankly incredible. So please do that and take a lot of time to go through this, because I think again, you won't have this opportunity in a lot of places.
Yeah. So one thing I can share is that we were wondering if the student doesn't have enough time to design and go for this design process, if us TA have the budget still for the semester, what should we use for? And I propose to make a lot of the colored proteins, and that's turned into the picture that you saw. So I order everything through Twist. I order it by making fragments because I think I'm going to be faster than them cloning, which is not true. We just spend a decent amount of time because it's not easy to clone. Some fluorescent protein doesn't want to clone because they're kind of toxic. But that's the reason we have a lot of things to do the art, and you see more when you go to the protein purification or other labs. So you can order and put into your vector. It's going to be useful. We're also going to have the custom vectors. So when you get into the fluorescent protein using in the next few weeks, you see how the vector that I make looks. So you can actually replace those fluorescent protein with the protein of your interest. So that would be another cheat code. If you don't know what to do, you just want to change the protein, you can just find my start codon, just replace that with your DNA sequence. That translates into the protein sequence of your interest. All right. That was awesome. I'll stop the sharing screen for now. And if there's any questions, I'll look into the Q&A. Do we have the Q&A on?
We do have the Q&A on. I can see if there's any.
I think most of the TAs are going to be on the other meeting though.
Yeah.
So most of the answers, most of the questions were answered already, I guess. So it looks like we are good.
It's good. I'll stop the recording here.
All right. Thank you, everyone.
What was the code for attendance today? Just nothing.
Oh, yeah.
Oh,
I just go to.
what's your favorite? What do you like? Plasmid or something?
Just put GFP in.
GFP. That's your code.
I would do most, is that fine?
Works for me.
Ronan, I have a question.
Yes.
So practically for lab time tomorrow, is there anything we should prepare beforehand?
If you come up with a design, that'll let you get started sooner, but you
Oh
can
yeah,
also, yeah, you can also design while you're there. So just please bring your laptop. That's probably the only important thing, actually.
great, thanks.
Still within the muddy drink time.
Oh, you know, which one is what?